Good day, everyone. Welcome to this important panel discussion organized by the Global Virus Network. I'm Dr. Maggie Lynn Bartlett, a virologist at Johns Hopkins Bloomberg School of Public Health and Program Director for the GVN. Thank you for joining on this crucial conversation on diagnostics for pandemic flu strains. Today, our panel is comprised of an esteemed group of experts who are going to help us understand the potential implications for human health and global health security as we strategize some responses to this viral threat. I'll now introduce our distinguished panel members. We have Dr. Erik Stemmy, who is the team lead for the Center of Excellence for Influenza Research and Response at the National Institute of Allergy and Infectious Diseases. His expertise includes managing a diverse research portfolio that investigates and delves into influenza pathogenesis, transmission, and evolution. We also have Dr. Andrew Pekos, who is a professor at Johns Hopkins Bloomberg School of Public Health, and he specializes in virus-host cell interactions of respiratory viruses and innate immunity. Dr. Martha Nelson is with us today. She is a specialist in pathogen evolution at the Human-Animal Interface at the National Institutes of Health in the US. We have Dr. Elise Stackler with us, who is a research scientist in the Participetti Lab at the Broad Institute and an expert in high-throughput CRISPR-Cas diagnostics. We also have Dr. Albert Osterhaus, who is a renowned virologist and professor at the University of Veterinary Medicine in Hanover, Germany, and known for his expertise in zoonotic viruses and contributions to global infectious diseases and pandemic preparedness. Dr. Philip Gauger is joining us from Iowa State University. He's a professor and an expert in diagnostics, as well as porcine viruses and swine production medicine. And before we go ahead and launch into this dynamic discussion, I just want to preface our talk today with a little overview of the three different key diagnostic methods we use for viruses. There are PCR-based tests that are a little bit like detectives when we're using a magnifying glass to find tiny clues. These can amplify fragments of viral DNA for accurate detection. We have rapid antigen tests that are similar to our home pregnancy tests or SARS-CoV-2 tests that can offer a fast and sensitive, suitable screen for initial screenings, but are not nearly as sensitive as PCR. And then, of course, there's next-gen sequencing, which is how we're able to distinguish variants. And it's a little bit like looking at the entire book of a virus's genetic code, where scientists can track the evolution and the mutations down to a single letter on a page. And now this continuous adaptation and improvement of methods is essential to meet the challenges that are posed by the evolving influenza viruses so that we can enhance our global pandemic preparedness. I'm going to go ahead and lead off the questions portion, and please feel free to submit questions that we will address to our panelists through the chat function or to Ruby. I'd like to know perhaps how the experts in this room perceive the evolution of our response to the current issues detected in cattle in the US compared to historical approaches in the field when we detect a new influenza strain. I might direct this first question at Ab, if you have comments.
Well, obviously, I'm not the obvious one to ask because I'm not involved in the United States. I'm in Europe at the moment. If you ask me about the historical part, of course, well, that goes for me back for a long time when we first discovered H5N1, basically in a young child who died in Hong Kong, and subsequent cases there, six of them died. It was altogether about 18. It was very difficult to convince the scientific community that this was really a virus spilling over from birds at that time. And when I talk about the techniques we had at that time, I'm talking about the last century, obviously. So basically, we used serology. We already used sequencing as well, but we showed indeed that we were dealing with an H5N1 virus that came from birds at the bird market, spilling over to humans. And when we sent our first paper to Nature, I remember that. It was not until we took Rob Webster on board, that actually we could convince the scientific community that this was really a pandemic threat. But I should immediately say that after that we have had a lot of cases of H5N1 in humans, even with high case fatality rates, which is not the case at the moment, obviously, as we'll discuss later. But there's always been the risk that this virus would actually spill over to humans and from human to human. And we did a lot of work in the lab in Rotterdam together with Ron Fouché and a couple of others, where we showed the number of mutations that were actually needed for this virus to become transmissible from ferret to ferret through either aerosols or small droplets. And so that gave us the idea that it was not more than a handful of mutations. So I think it's interesting at this moment when we're talking about the subject that this virus has a very low potential to be pathogenic for humans, but it probably would not need a lot of mutations of this virus to actually become a transmissible virus. And then transmissibility does not equal pandemic, but that is one of the conditions that's there. And of course, when we're talking H5N1, it's not one virus, obviously. So we're talking about all kinds. The virus that you find in cattle today is a completely different virus, but the hemagglutinin and the neuraminidase are quite similar. Yeah, and we have, of course, this new clade of viruses that's spreading around, coming from initially from Asia through Europe into North America, South America, and basically going globally. And this is really a virus or a group of viruses that has killed a lot of birds, a lot of mammals, and has now come very exceptionally into cattle. And I think that's an important thing to realize, that you always with influenza have to expect the unexpected. We were preparing for an H5N1 pandemic already in 97 of the last century. And then whilst we were gearing up, it actually became an H1N1 pandemic. So it's very difficult to predict, but it's very important with the techniques that we are talking about today to really follow exactly what's happening, see what species get infected and what mutations are accumulating.
And Maggie, maybe I can also talk a little bit about, one of the challenges that we're seeing is that testing is available. There's a pipeline for testing that's in place, but getting access to the actual samples and the populations that we want to test is really the gap here. The USDA, CDC, even the FDA, when it comes to milk products, they don't have the authority to go in and mandate testing. They're required to ask for volunteers, to look for input from in this case, cow farm owners, cow workers. And we've seen that there's a reticence in those populations right now to really participate in this. So again, I think that's very different from the COVID-19 pandemic, right? Where we didn't even have the tests initially, right? But people were wanting to test. We were a very different problem here in terms of how do you get access to those populations? What are the barriers that are keeping those individuals from wanting to participate in something that and I'm sure everybody on this panel would find is to be an incredibly important issue, which is to get an idea of the extent of exposures and the extent of risk.
You have a principal difference with what is happening in Europe. I think the national authorities and the European regulation allows you in their respective countries to close farms, to do much more in the veterinary field than I understand is possible in the US, which is really a very bad sign and makes it very difficult to control the disease. If you were to do the same with foot and mouth disease, I'm a veterinarian by training, so if you were to do the same with foot and mouth disease, you're not going to control it for sure.
Yeah, I think that's a really fascinating point to lead off on, and having it be kind of an opposite issue to where we were with COVID-19, and that we have the tests, but we need to get to the samples, and thinking about how we can mirror what's working in Europe in this case to what we could apply in the US. Perhaps these aren't in place just because it hadn't, we didn't have the surveillance that we have today until fairly recently. And so what ways, I wonder, yeah, Andy, go ahead. You look like you were going to say.
You were going down the pathway, and that, certainly there are ways that maybe mandating testing is one way to do this, but I've also thought about the fact that you mentioned point of care testing. Point of care testing is a way to give the power for the surveillance to the actual farmers, to the farm workers, and to put some level of control back into their hands. I think one of the issues that really does come through here is individuals are afraid to come forward for various reasons to be tested. I think farmers are afraid of having large number of people descend upon their farms, right, and do this testing. But if you had a point of care, for instance, and you can give a farmer 100 point of cares and say, here's what you have to do. Some of that expertise is on those farms. And that might be a way to try to get more testing in place, which I really think is the critical thing at this stage.
But you have to create an incentive for the farmers as well there, because
Yeah.
if they're only being punished afterward, they're not going to be very eager to do the testing.
Absolutely true. Absolutely true.
Yeah, and I wonder if that might be a nice time for us to, Elise, if you might want to speak on your diagnostic approaches with CRISPR-Cas9 and how that might be useful in these situations.
Yeah, so we really focus on two different formats of our CRISPR-Cas diagnostics. One, which is very high throughput, where we test for a lot of different pathogens from one sample source in a central lab, high throughput. And then the other that we have developed is really this point of care, point of need testing, where you could test on site. And so that's where I see this as being something that is potentially useful to deploy in the field.
And I wonder if there are thoughts that people might want to follow up on for these point of care ones. How sensitive are they? Are they able to distinguish between ones that we would be concerned about and ones that we're not concerned about? Or where are we at this stage?
In terms of variants, like
Yes,
strains?
yeah.
Yeah, so for the CRISPR diagnostics, we definitely have that capability of getting really good distinction between different strains. Actually, in our lab, we've developed multiple different tools to help with this. And our latest one, which I know is in preprint form, I think has maybe just been accepted as well. What it's really good about is distinguishing even a SNP difference in two different strains. And we actually have that in our lab already that we're using to test in terms of an H5N1 assay that's on target for this specific outbreak clade versus that H5 that is not part of this outbreak specific clade. There was a lot of discussion internally about what level of specificity we want to do. Like, do we want to test for just the cow specific outbreak? Do we want to test for the outbreak overall? I think one of the biggest concerns was, so we landed on testing for this specific outbreak overall. So that would include like birds showing up as positives. One of the things that we were concerned about is making it too specific and then losing that sensitivity in terms of like, then if you're looking for human cases, would it mutate enough that you wouldn't be detecting it depending on how narrow in you focus? So we took a little bit more of a conservative approach because you don't want your diagnostic tool to become irrelevant so quickly.
I think the problem you're facing at the moment in the US is to identify the positive farms there, and even with the non-specific assays, not at that level of specificity, but you really want to make a program where you can identify farms that are positive and close them off. Yeah, and then so it's better to go with a broad test first, and that could be any of the tests that have been mentioned. And then once you have identified the positive farm, then you go in more detail with the CRISPR-Cas or whatsoever, next generation sequencing or whatever, what have you. But the most important thing at the moment is to stem this thing, to make sure it doesn't leak into South America, Central America, and that it goes much wider than that. Fortunately, in Europe at the moment, we have no indications of a thing like this. So there is some monitoring ongoing. And I think the other thing we haven't mentioned yet is serological monitoring is quite important as well, of course. So that if you see what's happening at certain farms, then really in a certain area start to get looking whether indeed you might have serological evidence. And these are very simple techniques in principle, can very rapidly be done. But it's important to get the cooperation of the farmers, obviously.
I can speak a little bit. I can only really speak, I guess, from the animal side of the testing, what that's been going on. at the Iowa State Diagnostic Lab, and we're not known for being a state with a lot of dairy cows. But right now, we have at least 9 different farms that we know of that are infected. And there's been a lot of work taking place now, and I think that can be some good knowledge just from the standpoint of knowing that we're and other labs are working on a lot of serological tests and adapting those from what's currently available for use on the avian side and also on the swine side with our very general nucleoprotein specific ELISAs and looking at the different sample types in which we would be able to detect antibody. And it's such an unknown area on the bovine side because it was completely unexpected to find an avian or just an influenza A virus like this circulating in bovines, let alone it being an H5. And so once it came to the point where we kind of exhausted our other expected differentials on the list for these clinical cows, that was started off as kind of a mystery disease back in January and February, kind of came to the point where we maybe start looking outside of the box and testing for influenza in general, in which I think when we're talking PCR and a rapid test, we have some very good commercially made as well as in-house made PCRs that are targets for influenza A in general. And so we start there with a screening assay. Once we know that is positive, then we move to our additional tests that are available for characterization. And for the most part, we haven't used in those instances an antibody test in particular as we work through characterization. But if we're labs within our National Animal Laboratory Health Network, we have the H5 subtyping assay that's available. And then because of the avian influenza outbreak, we also have another specific 2.3.4.4b clade specific PCR assay available as well. And that did kind of come about, to my knowledge, I think because of concerns maybe that H5 assay might miss an H5 particular genotype, so to speak, or a mutated form of it. So the 2.3.4.4b was developed to be specific to this particular strain and clade. But once we do have the ability to use those assays through deviations on mammalian samples. And so we can choose to use those on what we want if we feel that it would be relevant. But once it's H5 positive, then, we do submit any and all of those samples to our National Veterinary Services laboratories, and they do the final confirmation. So those PCRs are available when it became obvious that this was circulating in our bovine species, and kind of some of that information got out and some of the floodgates opened, if you will. A lot of testing started to take place. And I think that's when we started to think collectively within the NAL network and other diagnostic labs and individual researchers, what other tests need to be developed for this purpose. And that's where we started to look into the antibody assays, particularly this NP ELISA, which appears to be working well, not only on serum, but also looking at it for testing in milk. And so I think that might be coming down the pike as far as an additional diagnostic test. And so long as we have our good commercial assays available for PCR, we can do a rapid test and then follow up with our H5 assay, so long as we continue to be able to do the follow-up sequencing as well. And that's where next-gen sequencing would come into play. NVSL is doing a lot of that sequencing. Most diagnostic labs and other labs have the capability so that we can monitor the variants that would be in circulation and really try to keep our specific assays like in H5 subtyping, where we're targeting a more variable region of the genome, keep those relevant so that we're just not missing an H5 in general, let alone if it were to be the highly pathogenic form. So those things are in progress. I think things are coming along well. I think so long as other labs can get support so that we can be ready to perform this testing on a high volume basis has become really important. We're just fortunate at Iowa State that on the swine side of things, where we do the majority of our testing, that particular industry has done a huge amount of surveillance for other pathogens that are of economic relevance to that industry. So that's kind of forced the issue to be able to do things on a high volume basis. Our extractions are on a 96 well basis, so is our testing. And then we can be prepared to run a lot of samples through in a short period of time. We had to do that for COVID testing for those diagnostic labs that converted over from animal testing to human testing at that time. we adapted our high volume testing methods to that particular testing as well. And so I think it's kind of proved its point, but I think what we're kind of looking at now is what do we do next for surveillance, especially in the bovine population that has not had to do that before. And, I'm on the human side, there's things going on as well. But again, like I said, my background is more on the animal side in testing. And the bovine side hasn't had to do that before for any of their particular diseases that they've had to deal with, unlike on the swine side. And so that would be the next thing that's coming. Mandates, whether they're going to be coming from the state governments as far as what testing needs to be done, particularly for animal movement. We do have, from a federal standpoint, movement testing that's required, that's in place. And in Iowa right now, we do have some circle testing that's going around avian facilities that have been deemed positive, looking within control zones at what dairy farms might be positive as well. And so through that testing, we've recognized many more farms because of those that particular approach. I think it's still unknown what's going to be coming next. And a lot of dairy farms are also maybe skeptical because they're not sure what could be coming next for requirements, how that might impact them too. So I think all of those things are on the table. I hope the surveillance continues. I hope we can continue to use a lot of next-gen sequencing to keep track of the variants and what's going on there. For me, in a diagnostic lab, I want to keep the tests relevant and have the knowledge that they're going to be highly sensitive and specific, that we can use them on a rapid basis and then cooperate with our federal labs in order to get them the samples they need in order to do the additional testing and the sequencing to make that available for what other monitoring needs to happen next. And for the bovine side, there's a lot, I think, of gaps. I think it's fair to say on the epi side, as far as how this got started, obviously it was in the cattle longer than what we realized. So a surveillance program that's ongoing seems to make sense because we're never going to be able to maybe catch these infections early enough if we don't have things like that in place. And that also is a challenge to implement. So that's putting a lot of comments in on a lot of different topics there. But from my perspective in the diagnostic world and testing in that aspect, that's some of the thought processes that are going on there. And I think going back to the serology comment, you will see antibody testing becoming more common now that those have been validated. Those commercial kits are already available. There just might be some upfront processing to different sample types other than serum than what we were used to using in order to get those implemented on a high volume and routine basis.
Thanks, Phil. I think you raised a lot of really important touchstones for us to circulate on. And Martha, I wonder if maybe you would comment a little bit about the penetration in sequencing depth in different species to date, because I know there's a lag in getting samples where they need to be, in diagnostics where they need to be. And at the beginning, there was a lag in getting the sequence data where it needs to be. And so we've had a lot of logistical hurdles in our response to this.
Yeah, sure, happy to. Yeah, so there's been an evolution in the way that the sequence production pipeline has operated. So right now, just the current state update is that USDA submits raw sequence reads to SRA, the sequence read archive, which is at NCBI. And those are occurring on a weekly rolling basis as these outbreaks occur. The caveat for those is that they have USA 2024, but they don't have a collection date, a month or date, or a US state. So you know the host, but you don't know much else about that sequence. So you can use that to maybe look at spillover into different hosts. You can look at mutations that arise, but you can't do the kind of classic phylogenetics of using genomic data to elucidate routes of transmission, spatial spread. We have these new outbreaks in Iowa and Minnesota. We can't really reconstruct where they came from and how the virus is spreading around the country. There are periodic so originally there was a kind of a plan that there would also be a 30-day delay. And after an SRA submission, those data would be published on GenBank with the collection date and the US state. and that would occur on a rolling basis as well. But those haven't really occurred since the last major submission that happened with the USDA preprint back in early May. So there's been kind of a drought in data for several months now. That makes it pretty much, from our side, we're just kind of flying blind.
Yeah, that sounds like a key challenge. And I wonder if maybe we could speculate on how to overcome these challenges when time becomes paramount to how all of us across the, virology field are able to respond to these sorts of threats.
I mean, if I can chime in, the work that Martha's talking about, right, is such a critical additional aspect to understand the transmission chains and the movement of the virus. When it comes to basic questions like how many times has this virus been introduced into cows, right? How is the spread going on? There's ways that you can look at the epidemiology from an aspect of the cows and the movement and those aspects of it. But the virus sequencing and that viral epidemiology, right, gives you an added layer that connects farms together and gives us a better understanding of, again, the start as well as the extent of this. Understanding that there may be some hurdles in terms of people not wanting too much granular information about this, but the lack of that, as Martha so clearly said, is really hindering a better understanding of how these viruses are spreading and where we need to be looking for to not only prevent transmission between cows, but also other incursions into the cow population.
I think one of the important things is also because you may potentially still be at the beginning of something that mushrooms, and what I think is important, and I think already Philip was leading to that we have different layers there, and so you need rough testing first of all to get a rough idea of what's going on in the country as a whole. Yeah, and so I was talking about serology that's apparently forthcoming. We are starting that in Europe as well, but also like we did for SARS-CoV-2, it's sewage screening. Yeah, for cattle it should be slightly different, but also milk screening, and do that at a much larger scale with generic assays. And once you have the positive ones, then home in, and then you can start asking the questions that were just addressed in terms of where does it come from, how is it spreading, etc. But first of all, you want to have a global idea of what's going on in the country as a whole. So I think the things like serology screening and also PCR screening of collections of sewage or whatever manure sources you can get. And so in that way, and then also the milk screening apparently is an important thing, not only for public health. And let's hope that all the pasteurization is going to be completely effective. But so I think you have possibilities to do that. And before homing in on all these very much more sophisticated assays, I think that's very important for the second stage. But the first stage is really to try and control the problem as a whole.
I think that's a really salient point, and one that, raises this issue of needing surveillance globally, of serum, of wastewater, so that we can get a pulse of what's out there and stay ahead instead of what we currently do, which is identify and then try to solve it once it's already out in the wild.
When we had the era of, for instance, TB screening, yeah, so which is not happening more at that scale. But then you already had a repository of samples from all over the place, and that makes it very easy to home in on the weak spots. And
I will say, in terms of overall surveillance and screening, milk and pasteurized commercial milk is sort of akin to wastewater screening for this outbreak. The biggest challenge there, though, is just understanding when you get a bottle of milk that tests positive, where is that actually coming from? It's very convoluted in terms of, you're not getting a sample of milk from a farm, you're getting a sample of milk from a processing plant that might have several farms that are feeding into it. And so it becomes much more difficult to understand than just like typical wastewater surveillance in terms of like COVID, for instance.
The milk is being transported from a farm to a central point. Yeah, so you could at every farm, in fact, before the milk goes into the general tank. Yeah, so take one sample, and if you do that, it's not a difficult thing to do. If you wait until it's in the bottles or in the cartons in the shop, of course, then you have another problem. But do it at the source, yeah, do it at the farms, yeah.
Absolutely, ideally you would do that. It then becomes convoluted in terms of you need the farms to participate.
Exactly.
Whereas for wastewater screening and sampling, people like that because people don't have to give their consent, which gets into a lot of other ethical issues, which is out of the scope of this panel. But if it worked the same way for milk, then you don't have as many of these issues. But because of this, you really do need that participation on a voluntary basis from the farms. Yeah.
But sewage has the extra problem, of course, that you might have material from birds as well. Yeah, so it's not that easy. Yeah. But if you take milk samples from certain farms, and well, at least in Europe, it has been and is being done for other purposes. So, but it's just a matter of logistics, and on our end, we can develop the most sophisticated techniques to answer the most sophisticated questions. But we need to have the basics first, I'm afraid. And I all the things about where does it come from, how is it spreading, etc. It's very important. Yeah, and also the pathogenesis. And is it the milking cups, or what are we really dealing with? But first, have to know where you focus and take your general measures.
Well,
Yeah, one thing. Oh.
no, go ahead, Elise.
One other thing I would like to add, so my background is in environmental engineering, and so I think a lot about, surveilling the environment as well. And again, like if we can get partnerships with these farms, what I would really like to see as well is a lot more environmental swabbing. And like, is it staying, on the milking equipment that is then being shared between farms? How can we give recommendations for how to disinfect before these things, or how long does equipment need to be held before being transported and used on a different cow? I would love to see all of that happen as well.
I hope that we're going to see a lot of good research coming out of some of this that's going to be supported by some governmental funding in Iowa, just down even to the state level. There's a fair amount of funding that's coming forward, and I think those questions, Elise, that you mentioned, as far as a lot of different samples being collected from the environment, from different risk areas that could represent where transmission is occurring, all of that's going to be taking place. So it'll take some time, obviously, to get the results out and to understand all what's going on. But I still think that we have some prototypes in place that have done well from a surveillance standpoint. I would really look to the USDA influenza surveillance stream that takes place in pigs at the moment, why we couldn't adapt that if funding was available on the bovine side. And really a lot of that is driven by an anonymous surveillance stream. From the pig standpoint, we can get in a lot of samples applicable to detecting flu. From the bovine side, maybe that hasn't always been routine, but yet there is a fair amount of mastitis testing and culturing that goes on with milk samples that come in. And for our diagnostic lab on the swine side, we can pull those samples into the anonymous stream, which creates confidence for the producer because their information, at least beyond the state level, gets stripped from the sample itself. So we know where it originated down to the state level at least. But then it goes through a process and we get virus isolates to use for challenge studies and vaccine production. And we also get the sequencing, which is really important. And then that can be used and is put out there into the public domain for a lot of research and work that has been accomplished. What's great from the bovine industry standpoint is we already have a routine population sample in place, and that is the bulk tank. And Albert, you were commenting on that as well as far as collecting and testing. Farms do want to have confidence in what their results will be used for before they really submit to doing that on a routine basis. However, there's other avenues. To my limited knowledge, I thought the milk processors, when they pick up the bulk tank samples, that they are required to keep a specimen of that per each tank or load set aside. That maybe would be an opportunity for some anonymous testing that could take place. And then you still are having it to the farm level and not having it mixed in from multiple farms, things of that nature. So maybe it's time to be creative and look at where some additional testing and surveillance streams could be adapted to the bovine industry. And we could look at this a lot more closely and further, and maybe In fact, on the SEER side of things that Erik's involved with, I assume that there's some optional funding that probably is being sought after by our SEER centers in order to look at some of those questions as well. So I think there's opportunity. We've got a surveillance prototype system in place on the swine side. Seems to be working well as an anonymous stream. Maybe don't need to know all of the details of the farm, but we get to take advantage of the surveillance, especially down to the sequencing level, and do a lot better job of monitoring and understanding what could be circulating out there before we find it after the fact.
Yeah, I think those are really important points, Phil. And Erik, I wonder if maybe you might take this moment to speak a little bit from SEER's perspective and the things that you've seen change over time in response to everything that we're finding out about this strain in cattle.
Yeah, sure. So the SEER network is a longstanding program with NIA that's been around in various forms since about 2007. So really, this is the type of thing that this network was created for and to respond to. That goes along with NIAID's dual mandate to maintain research and also respond to emerging and re-emerging threats. And so one of the ways we do that, right, especially in this case, was to leverage the things that we had ongoing at the time when the virus was first detected, and to do some of that early on the ground work that we were able to access. And then as that progressed, we were able to transition it to our other US government agency partners that have more mandates and other responsibilities in other areas. And so I think that's one of the things that was really useful, or especially early on in the outbreak. And so, Phil's right, we are in the process of trying to work with an internal risk assessment pipeline that we have established through the SEER network to understand what research is ongoing within our network, what research is going on globally, and where we can fit in and gap fill within those areas as well.
Yeah, I
I
think
have,
that's
I have
go
a
ahead.
question about a subject has not been brought up, but what in regard or looking at other ruminant species like small ruminants, so sheep and goat, is there because there have been some minor reports about that. Is that an additional risk?
Well, we've had some publications related to goats in Minnesota that have been confirmed to be infected with H5, and one with this strain. And so I think in what knowledge we have of this strain so far, it makes you wonder if there are a lot of different species on the mammalian side that are at risk. A lot of that has just come out from the fact that this strain has persisted. We saw a lot of birds that were affected outside of traditional waterfowl, and that seemed to be a little unusual. And then we started to see back a year ago with some things that have been published, a predilection for mammalian infections as well. I think it started in the wildlife, which would make sense for what exposures could take place. I know Iowa State's diagnostic lab and multiple others in the US had wildlife submitted that had very classic lesions for H5, highly pathogenic avian influenza virus. You can sequence from all of those different sample types. And when you talk about when you get into the world of pathology, what is an additional supporting diagnostic tool is just in situ hybridization and immunohistochemistry that's supportive as well to help really, I think, solidify where these infections are taking place and what tissue tropisms are occurring with these particular strains. And so that's getting beyond PCR and all these other techniques that we have that are good too. So there just definitely is a mammalian predilection for with this virus this time. I don't want to mislead anyone and say that there's particular species involved, because we've tried to be careful within our realm of talking about the bovine cases that is there something about this virus that is bovine specific? And we really don't know that at this time, and don't think that there is necessarily, but certainly from a mammalian standpoint, there is. So what species would not be at risk? I guess it would be those that definitely wouldn't have some type of risk of exposure and potential infection for it. I guess we just don't expect some species to be infected, and bovine certainly was the situation, and that has such a predilection for the mammary tissue, but some of that can make sense to the standpoint of the tissue that would be represented there and where the viral receptors are located. Just maybe means we need to think outside of the box a little bit more and where else we could find this. And of course, it's hard to talk about flu without talking about pigs. And there's research being done that's already been done and published using these strains of H5N1. There's probably going to be more coming, especially with bovine specific strains that have been isolated from the dairy cattle. But using mammalian strains from fox and some other species, they've been put into pigs by our USDA collaborators, and they can become infected. Thankfully, right now, it doesn't look like that there is a great propensity for transmission from pig to pig. But we understand on the pig side how our endemic strains are common. Those would be the H1s and the H3s. And if pigs are endemically infected, they're going to have those strains present at the same time they could be infected with an H5. And so then you get into the topic of reassortment. It's very real, and I think that's the risk that we're concerned about there, as far as what could happen within pigs that are implicated as a mixing vessel. They're certainly not the only species that are, I think is safe to say. But what could happen when we have reassortment with endemic strains and picking up those internal gene segments that are going to be swine related, and then we know that people and pigs can share those strains as well, and what that could mean next. So hopefully there will be some research taking place there as well, but a number of mammals that could be potentially at risk for these strains with what we, I guess, have garnered for the information and research so far, and maybe what that could mean for focusing research next, and really for thinking outside of the box when we're getting in these submissions to animal diagnostic labs that aren't fitting the typical scenario for what we think differentials should be for clinical signs, and be using these tests that are available to look into it further for flu, potentially for flu H5.
We were involved in the early detection of virus in encephalitis cases of especially seals, the H5N8, H5N1. And that spilled over directly from birds. That's what we think. My question was more related to our specific measures being taken for milk coming from sheep and goats, and especially in Europe, unpasteurized milk for cheese production is very popular. So that will be an additional risk. I take it that's being taken care of. Yeah?
I don't know if I would assume I hope it's being taken care of, but I'm with you that I hope it is, but I'm not sure. You know, if it was a surprise, then maybe that's not in place yet. I'm not sure if anyone on this call has insight into that.
I do know that the FDA has discussed an expansion of their current testing from just milk products into cheeses and other items as well to I think get at Ab's point, which is, you know, unpasteurized milk does go through certain processes for certain cheeses and those kind of things, and that represents another unknown risk in terms of potential transmission. But I also want to get back to you know, Philip's point about you know, the risk to mammals. I mean, it's important to understand this is really an unprecedented period of time for the H5N1 virus to be continuously moving through mammalian hosts. Many on this call and others have seen really interesting mutations in what we think are one-off infections of mammals, right? And we see signs of adaptation of the virus to mammals. This is now a situation where we have extended movement of this virus solely through mammals. And so, you know, getting back to the point that Martha talked about, the only real way to understand some of these sequences is to really also have a little bit more information about the timing and the location of these, so that we can really understand where are the linear pathways, what are the hotspots that are changing here. If this is primarily a transmission through milking equipment, that may be a very different pressure on the virus than a transmission through the respiratory route. Hopefully that's a little less risky in terms of the pandemic potential, but you know, we don't know because we've never seen influenza virus do this kind of transmission through mammograms for this extended period of time. So again, just so many questions that that the data is sort of around, but needs to be sort of curated a little bit more and more granularity put onto it so that we can really move on to some of the other basic research programs that can really assess the greater risk to the population of these kind of outbreaks.
Now, it's interesting that a few human cases they were related indeed to exposure to milk, yeah, or at least the milking process. Yeah, so that already shows that the respiratory transmission is probably not the most frequent one.
Yeah, I think it's a interesting aspect of how we're addressing this issue. And I wonder if we might take this opportunity to look at the questions that are in the chat. I'm not sure if anyone's aware of if H5N1 is still select agent. Ray Sinazi is asking, or if BSL-3, or what sort of regulations are in place. And now that we're seeing more, do we anticipate changes in that?
I think the current status of H5 is that it is still considered to be a BSL-3 level pathogen. It has been taken off of the select agent list, but there are still specific permits and there's still some controls in terms of who can obtain those samples and what levels they'd have to be worked on. But I think because of the extent of the presence of this virus in so many products, that movement off the select agent list has been quite helpful in terms of allowing multiple laboratories to really do some more experiments with these viruses. So I think that's been a good move. Again, I think safety is still being put in place because of the maintenance of this virus at BSL-3 levels, but the removal of the select agent status certainly does help with the research efforts on these group of viruses.
Let's say BSL-2, BSL-3 restrictions for influenza viruses, of course, is a difficult thing with all the human viruses, and still work with H2N2 ongoing. So it's a very difficult thing. And if you look at US, it may be more clear, but in Europe, it's quite patchy, and there's different regulations in different countries also. But I'm not so worried about spilling out of labs when as a matter of fact, we have a big problem in cattle at the moment. And of course, it's important to have safe labs, but talking about protecting viruses from escaping from the lab is not the primary issue we should think about at the moment.
Yeah, I would completely echo that sentiment. I think this highlights just how many of these viruses are in animals that we interface with on a regular basis, and that, you know, we're surrounded by them. And I think this crowd is very aware of that being the predominant method for viruses getting into people. I do see there's a chat about vaccinating high-risk individuals and dairy and cattle workers. I haven't heard too much about this, if this has been pushed forward. I'm not sure if anyone on this call has comments, or if we should contemplate incorporating this vaccine into this year's seasonal influenza. I'm not sure if that's been looked at.
I think that would be too premature, but I think we should be ready for, let's say, in our pandemic preparedness. Although the risk is low, the risk is not zero, and a low risk for something that's horrible that should be taken into account as well. So we should have the candidates for vaccine production ready for humans as well, but to incorporate it in the normal human seasonal influenza vaccination, I think that's not the question. But I remember that when we had the early H5 problems, H5N1 problems in Southeast Asia, I was involved in actually running clinical trials. Yeah, also organized by industry for H5N1 in some cases, even human clinical vaccination of humans ongoing in Taiwan, for instance. Yeah, so it should be taken serious, and we should keep an eye on that, and we should be prepared, especially after what happened in the swine flu, the Mexican swine flu, or what we have seen happening with SARS-CoV-2. So we can speculating that this is going to. We all speculated that H5N1 at that time was going to be the next pandemic, and it was H1N1. So it's very difficult to predict, and so there are candidate vaccines, of course, and they are available, but they have to be scaled up if really the worst thing happens.
But I think it's important to also note that vaccines can be used in multiple ways. And one of the ways could be to find those high-risk populations right now and provide them with the vaccines to do a preventative sort of pre-pandemic, another wall of defense to prevent that virus from infecting humans. I think that's something that could be considered. I actually don't know how seriously that is being considered, but I know some of the H5N1 vaccine stocks that are present here in the US, very limited amounts in terms of nowhere close to being able to be used to immunize the population. But I think at this stage of the epidemic, we need to think more about the strategic use of those to protect those vulnerable populations, those high-risk populations, like the dairy farm workers. That might be some place that the current vaccine stocks could be very, very useful. Now, the problem, of course, is you can't get those individuals to really provide samples for testing. I'm not sure how willing those individuals would be to take a new vaccine based on everything that we've seen with COVID vaccines and those kind of things. So I think there would be a place for strategic vaccination right now, but again, a lot of logistic problems in terms of implementing that effectively.
With the vaccination that we were doing in Taiwan, actually, that was the healthcare workers. So that was a very motivated population at that time. With cattle workers, I'm not sure if that's the same thing.
I think in some cases there's even been a struggle with encouraging PPE use, right, in some of these areas. And so I think, there's probably going to need to be a large outreach and kind of educational campaign if things change and the need for vaccination becomes more pronounced.
Yeah, I think the first priority at the moment, obviously, is to control it at the site where the problem is really now with the cattle and the milking procedures, etc., and then identifying the positive farms and try to reduce the problem. And I think perhaps as well as the way we are looking at it from a European standpoint, we are a little bit surprised that in a country so well organized as the US, this cannot be controlled easier. And I'm just wondering what would happen if you would have an outbreak of foot and mouth disease. Yeah. Then if you deal with that in the same way, I said it before, but if you deal with that in the same way, you're not going to control that. And I'm specifically looking at Philip. Yeah. So basically, if you get African swine fever in, yeah, so you will be worried about that. If you were to handle that in this way, this is not going to stop, for God's sake. And given the ramifications towards the threat to humans at the end of the day, albeit little, then still I think we, at least in the US, you would have, I would suppose that you should have your act together better than you have it today, I think. So really try to limit it and push it back rather than seeing it spreading from state to state at the moment. But it's easy to say that from a Europe perspective. I realize that.
I think Phil can comment on this more, but one of the, Phil and I have both worked with influenza and swine and to some extent poultry for a long time. And I think one of the things that we've seen is how, comparatively well organized swine and even poultry are, and they have a lot of experience combating influenza, building these surveillance pipelines, sharing data, developing vaccines, and that in some ways, cattle have been kind of buffered for so many decades from, not just influenza, but also blue tongue and rinderpest and, other things, foot and mouth disease and other things that, have sort of shaped biosecurity in cattle in other continents. And this is kind of a first taste, and there's kind of a very steep learning curve that we're going through right now.
Yeah, so you don't have the problems with blue tongue at the moment like we have them in Europe.
Exactly.
That already creates, so you have to have a good system, otherwise you're not going to control it.
Right, but in some ways that system gets built kind of uncomfortably during crises.
I agree. So our BSE crisis, that was also, we learned a lot. Identification of animals, all these kind of things which didn't exist before. Every cow in Europe and every ruminant in Europe has an ear tag, and it's being followed very, very intensely. Yeah, but I think what you said is quite important. That let's say the avian and the pig world worlds are much more advanced than the bovine world, and perhaps it would be good to get together, to get to get the experts together and see what you can learn from each other. Although the logistics is quite different, of course.
The cultures are very different.
The cultures are very different as well. But I think the power of the farmers, so I hear all the time, so anonymized and all these kind of things. Yeah. And so in Europe we have all these compensation schedules, etc. And I'm not sure how that works in the US, but which can be counterproductive as well. Of course, in a foot-and-mouth outbreak, if you get compensated, there might be an incentive for farmers to spread the virus by themselves. So you have to be careful there as well. Yeah. So this is, as you say, it's cultural, but it's also historically grown. Yeah.
Yeah, and I think it really took everyone by surprise to see as far as this was circulating in cattle in the US. And, I think perhaps there was a little bit of, well, it'll resolve. And as more and more data came out, we saw just how vast it was. And I think adapting and getting people together from other animals of interest would be helpful. I noticed there is a question in the chat about the role of mice in transmission within farms, and I'm not sure if that's been looked at in this case, or if that's something that's considered for this.
There is a report on mice. I haven't seen it formally published yet, but there's some discussion about that. Yeah. Who knows that?
There's only one mouse sequence available, so.
Yeah. But so that's probably not the main route, man.
No, it looks like it's just spillover from cattle to mice.
Yeah.
But and I do see Peter.
For me, it's scary, some of those cats drinking raw milk. Yeah. So that's a little bit reminiscent of what we see in the seals. They get an encephalitis and die before they get into the lungs even. Yeah. So this is pretty acute. Yeah? And it tells you something about potential exposure to milk, to unpasteurized milk, what the consequence of that might be. And the important thing, of course, there was the paper last week by Vincent Munster's group about the conditions of pasteurization in the New England Journal, and yeah, I think it's quite important to make sure that all these plants that they really comply with the requirements there. That not properly pasteurized milk slips into the system.
Absolutely. And I think, this has been a concern in how far spread this event will occur. Peter Openshaw just shared a link for a UK news article about flu vaccines starting to be stockpiled as, the world is watching how we're responding to this issue.
Well, asking the question is answering it, I think. Yeah. So definitely we should at least have our seed strains ready and also the production facilities ready. And we don't know if it's going to be H5N1. Yeah. So the bovine story took us by surprise. So none of the people working in flu would have ever thought that a thing like this in ruminants would happen. Although we knew that theoretically you could infect cattle with some influenza viruses, but this was never expected. So in flu, the things always happen. So you can stockpile an H5 vaccine as we started to do before, and then you get an H1N1 pandemic. Yeah? So, but I think what you should do, of course, is have the seed strains ready in principle, or this whatever platform you're going to use, to have it ready to scale up very fast. Well, we started with diagnostics, but I think we're discussing a lot more than diagnostics.
Well, I think we've uncovered, that this is quite a complex issue, and while we have diagnostics, we don't have the logistics really in place for the issue that we're facing right now. And figuring out how to smooth out those logistics so that we can get a sense and solve some of these problems seems like a key issue for us to push.
Absolutely. So I think on the other hand, I was bashing quite a little bit on the reaction of the Americans. On the other hand, I think all the sequencing, etc., that's going on in the US, of course, is exemplary, and so I think we should recognize that as well. So that is a fantastic thing, and so and also the way that has been organized in the meantime. And I think
Yeah.
we're missing a similar system in Europe at the moment.
But as many have mentioned on this call, I think this is a great time to be also a bit reflective, right? In terms of, how can we overcome some of these hurdles that are in place in terms of access to samples, information from samples to get a better understanding of this. We could probably look to emergence of this virus and or any virus in a different scenario, but the same principles of you have to have the testing, you have to have the access to the samples, and you have to have the ability to do that in-depth tracking as soon as possible if you have any hope of stemming a wider outbreak. And so again, understanding maybe more incentives, right, to have farmers test should be more broad scale. There are things that we can think about now in terms of our pandemic response planning that is good to have some time to think about that now.
Yeah, and also the possibilities to enforce certain things on farmers, so because if that is not possible, and I understand that's a sensitive issue, but if animal health and public health is at stake, I think there should be pushed for. And I think what they understand is in the swine world is much better organized, and and also for poultry farms, etc. I think it may be easier than with cattle.
Yeah, but I think that hits on the, the growing pains of adding that to be something that's done on bovine farms. And Ray, thanks for this press announcement about an over-the-counter point-of-care test for influenza and SARS-CoV-2. It's things like this. Can we distribute these to people who have good connections with farms and let them know they can test and coordinate how that gets back to being sequenced without there being fear of repercussions. Of course, someone has to come up with the incentives to make sure that this is going to be done regularly, and we'll need modelers to determine how often they have to test, or if it's just a weekly swab of the milking equipment. I think these things haven't quite been worked out.
Yeah, I think in the poultry world, it's better organized in terms if you have an egg drop or whatsoever production drop, you see it immediately through all the systems. Whereas for cattle, it's not so well monitored. Although in the milking farms, it's getting more routine as well.
And I see one other comment in the chat. I'm not sure about if butchers have been tested. That was frequently done during the SARS-CoV-2 pandemic, but are butchers of cattle being surveyed for serum or exposure?
My knowledge, that really, as far as the beef cattle side, that has not been looked into to any degree, and probably because there hasn't been clinical signs that have implicated much of anything. There hasn't been really the testing taking place, and actually we have, other than just the beef cattle, the feedlot industry, we've got beef cows that are on pasture now that are lactating, and they've got baby calves aside, and so it does make you wonder if that species could be affected as well or infected. Maybe there could be a little bit of a lag of an area that we should maybe be looking into. But until we have clinical signs and a reason to test, we don't always get those samples as well, just like the comments earlier that have been. So for now, not to my knowledge, until something were to change from the clinical side of things, before we start looking there, will that take place, unless maybe some research might be being planned in the beef cattle side just to understand that better, in addition to what's going on in the dairy cattle.
But if the transmission is really going through the milking machines and the suction cups, etc., so it will be individual calves that will be infected. So that will not cause a major problem, I think. So it will not spread amongst a beef cattle farm. So I don't think that will be a high priority. I think people on the slaughter line, yeah, people slaughtering cattle on the slaughter line, but slaughtering dairy cattle would be a different story, of course.
So that's what we believe now as far as the transmission goes, and why maybe it would be more practical to look at the beef cow side, the lactating animals there, to see if they really would either be at risk or present a potential risk for it, as much as anything that's non-lactating or our feedlot cattle and things like that. That's true. So
yeah, and cattle that are being slaughtered, usually they're not lactating anymore. So but people who are doing the milking, you know, so the milkers, the people working with the milking machines, etc., they could be checked if they have respiratory symptoms, or even if you would do serology there, that would be quite interesting, of course. But there's so many things you can organize at this moment. The most important thing is to stem the whole thing and to see if you can get it under control. And the other thing the other thing we don't know is if you look at the plethora of different avian influenza viruses, we're talking about H5, yeah, but we know that if you look at the seals, for instance, or the terrestrial mammals, then you see there's a lot of other viruses that can cross the species barrier. And so you have the H10s, and in humans you have H9, H7, H what have you, yeah. So there's a lot more than only H5, and I think we should have a little bit of a broader mind once this is under control. That we have a system by which we can look at this a little bit more extensively.
Yeah, I agree. I think the disease X perspective is highlighted more and more. You know, we see a potential threat, but that doesn't mean that if we turn around, we're not going to see 10 others that have equal risk, as well as balancing what the real risk is and what sort of mitigation measures we can put in place. Sten put in the chat an interesting comment about another virus family, arboviruses, which have very different responses between Cuba and Puerto Rico. And so thinking about other ways in which other countries are handling their response to those type of threats and how we might apply those more broadly and have a broad viral surveillance and detection pathway. I think we often, and I welcome any comments on this, get siloed into our viral families, and then but they're all co-circulating in different places and all have potential for
well, but there are good programs for foot and mouth, and we mentioned blue tongue, and there's a number of these things that we have very good programs for, at least in Europe at the moment, yeah, because we get the threat constantly from Africa, and so many of these things are moving up. So also the flavivirus, etc. But I think you can learn a lot from things that are extremely expansive, like foot and mouth disease. Yeah. So see what kind of measures are being taken there. I'm not just talking about the diagnostic measure, but also what legislation can be made and how you can, at a certain point, if it's very important, how you can force samples to be taken, etc.
Yeah, I think that's a very good point to maybe end on. If there are final comments, I think we've, circled the issues with diagnostics, and that the issue really isn't with the diagnostics for this specific virus, but the issue with getting the samples to where they can be tested and then getting that data to the people who need to be analyzing it, and then the logistics and the incentives. And so it's a expansive issue, but I think that's a lot of opportunity for the US in particular to really improve its pandemic resiliency, now that we see this potential gap that we had open. And I want to thank all of the panelists today. This was a really engaging discussion, and I want to thank everyone who submitted questions. If there's anyone who joined us late or would like to revisit any part of today's panel, we will be making the recording available shortly. And then again, thank you all for joining. Thank you to each of the panelists. I really enjoyed getting to have this discussion with you. We look forward to getting to see you at future events. Our next panel will be July 30th at 3 p.m. Eastern Daylight Time, and the topic will be announced closer to the date. And this will go ahead and conclude our panel on influenza. And I encourage you all to stay persistent, resilient, and innovating faster than viruses mutate. Thank you all.